Good morning. It's 9 o'clock here in San Diego, so we'll begin the webinar. Today's webinar is on covalent ligand docking. My name is Andrew Ori, and if you have any questions after the webinar, please feel free to email me or call. During the webinar, please use the chat option in the GoToMeeting dialog box and ask your questions. I'll try to get to them maybe during the webinar or afterwards, or via email afterwards as well. All participants receive a 30-day ICM-Pro license. I will send information about how to get that in an email after the webinar if you don't already have the license. And you can follow the webinars from the past webinars on YouTube, and you can follow ICM News in Facebook and LinkedIn, Twitter. As I mentioned, last week's webinar, there's been 3 so far on ligand docking and screening and induced fit docking, and you can view all those on our YouTube site. And some of the setup for docking that we're going to cover today is covered in the first webinar called ligand docking. So the topics are how to set up covalent docking in ICM-Pro, how to sketch and apply a covalent docking reaction mechanism, and then once you've sketched it, how to run the covalent docking and screening using the docking menu. And finally, we'll show how to optimize and design new covalent bound ligands using the ICM-3D Ligand Editor. So today we're talking about covalent ligands, covalent inhibitors, covalent drugs. There's always been a reluctance in some cases to add an electrophile to a drug for concerns about inhibitor toxicity and other risks. However, there are actually more than 40 FDA-approved covalent drugs on the market. And there's an excellent review here by Renata Bauer, who describes the history of covalent drugs from obviously the early ones, aspirin and penicillin, through to omeprazole, the proton pump inhibitor, and Plavix, the anticoagulation drug. So if you do go down the covalent route for drug design, there are some advantages for producing what we call maybe a targeted covalent inhibitor. So one of them would be potency. So covalent bonding can lead to potencies and ligand efficiencies that are quite exceptionally high. Covalent bonding allows the high potency to be achieved in compounds with relatively low molecular mass, for example. And another advantage would be selectivity. So covalent inhibitors can be designed to target a nucleophile that is unique or rare across a protein family. And so this approach can lead to high selectivity against closely related proteins, because although the inhibitor might bind transiently to the active site, it will not covalently label them. So you're actually targeting maybe a cysteine residue or a serine that's unique to a particular member of the family. In terms of pharmacodynamics, the restoration of activity after a covalent inhibitor binds irreversibly requires resynthesis of the protein target, and therefore it's advantageous for drug pharmacodynamics and things like level of frequency and dosing effects and stuff like that. And also a covalent inhibitor could also be used as a biomarker to assess like target engagement in preclinical or clinical trials, in clinical use, to look at the relationship between the dose of a drug and the efficacy or toxicity of that drug. So ICM has all the tools that you need in order to design structurally a targeted covalent inhibitor. So as I mentioned, covalent complexes are an important class. However, the method for docking requires a little bit of modification in order to handle covalent bonding. The reaction describing transformation of you need to describe a reaction. So here we have the boronic acid, and it's covalently binding to a serine. So we have the serine atoms here. So here's the boronic acid and this drug, and so it's going to covalently bond to the serine residue here in the receptor. So we have to describe the free ligand before docking and a product comprising of the covalently linked ligand and receptor side chain. The side chain needs to include the atoms described are described in the grid, but are masked in the grid, so you don't see those. And input ligands are automatically transformed, and the designated side chain atoms are constrained during the bias probability Monte Carlo simulation that we use in ICM. As an example of the performance, there's an excellent benchmark published in the Nature Chemical Biology by Ligand Editor, and it's a benchmark that includes 61 complexes of beta-lactamases, penicillin binding proteins, and boronic acid and beta-lactam inhibitors. And ICM outperformed all the other methods by a very large margin in terms of average RMSD. So our MolSoft RMSD was 1.66 versus 2.87 for DOC, 3.4 for covalent DOC, and 3.5 for auto DOC, and 4 for gold. So how do we do this in ICM? We need to first load the ICM software, and I'm going to give an example. We have with the penicillin binding protein 3, PBP3, it has the PDB code of 3 PBS. So you just go to the search tab, type PDB search, and press enter. This loads the structure. I've already converted it to an ICM object. You see it says ICM here, so it's already had the hydrogens added and some conversion processes involving histidines and sparagenes and glutamines, which we described in the first webinar. But when you read it, it'll be an X-ray structure, and you right-click and you say convert to ICM object. So this is an important, it's an essential enzyme responsible for the final steps in peptidoglycan synthesis, which is a major component of cell walls. And it's covalently inactivated by beta-lactam antibiotics. So, and one of those is bound to the structure. So we can look at the interactions between this drug and the receptor by just right-clicking on pocket and say ligand pocket interactions. And then if we just double-click on the ligand, and we can just zoom in, okay. And we can see that there is a number of hydrogen bonds between the ligand in yellow and the receptor in blue, and also this covalent bond here with serine 294. So in this example, we're just going to redock this ligand. So first we need to move the ligand out of the pocket. Otherwise, it'll be seen in the maps and you won't see any cavity here. So we right-click on the ligand and choose extract ligand, move from object, sorry, move from object. Now we have two independent objects. They don't see each other. We have a gap here. So now we need to set up the docking project like we did in the first webinar. So we go to docking again, docking menu, go to new project, and give the project a name. And ask you for the location where you want to save your project, your docking project files, and the receptor object. The receptor object is 3PBS, so it finds it automatically. Then you could run ICM Pocket Finder to find a pocket, or you could just select some residues in the pocket. But we know the ligand is in the pocket, so we can just double-click on the ligand and choose the option Define site around selected molecules. You can see it selects the residues surrounding the ligand. And there's an option here to say make receptor maps. If you were doing a regular docking, you would leave that checked, but we're doing covalent docking, so we need to remake the maps in a minute. So I'm just going to uncheck that box and just go OK. And we start once again with this probe, which would be where the docking begins. And so it's usually in the center of the pocket, so you can just leave it there. Just press go here. And then the purple box represents the area where the maps will be built when we eventually make them. So you can make this bigger or smaller, depending. But usually the box is of a decent size to encompass the whole pocket. So just go. Okay, so now we need to tell ICM that we're going to do covalent docking. So we need to go to the docking menu and go to covalent. And the first thing it asks us which residue is going to be modified. So which residue is going to make the covalent bond. And in our case, it's going to be that serine 294. So I need to actually go back and select that serine 294. So one way to do that would be and it needs to be selected in this new object that we've created for docking, which is the name of the docking project _REC. So we need to select serine 294 from this structure. I'm going to undisplay the other stuff just for clarity. So we can do that by extracting here, expanding here, and choosing and finding the serine 294. So you select it. You can see the selection is blue in the ICM workspace, and we have a green selection here. So that means if I press center, it will zoom into that residue. If I do other things like press residue numbering, labeling, it will label that residue as well. So we made our selection. So then we go back to that docking covalent option. You see now that we have one selection, so one residue selected, one modified residue. And so the next step is to select the reaction that we're going to use in order to build the covalent bond. So we're going to go to load preset reactions, which is down here. And this is a set of reactions which I'll show you in a bit that we've pre-drawn and created for a number of common covalent inhibitors. And I'll show if your mechanism is not included, I'll show you in a bit how to sketch these things. So we're trying to find the reaction that we need is this beta-lactam reaction. So we just click on the reaction here, and we need to choose add to list. So just click here. Now we can see the reaction shown here. You could have more than one reaction in this list if you have different chemistry in your structures that you're going to dock. So now we go OK. We'll remake, or in our case, we're actually going to make the receptor maps because we didn't make them earlier. So we just click here, go OK. And this is making the maps, and you see it's modified the side chain of this serine. It takes a little bit of time to run, so it's completed now. So the next step is to dock. And we're going to dock. There's no point trying to dock the ligand that's bound because it's already in the reactive state. We need to draw it as it is before the covalent bond is formed. So we go to the ligand sketch option here, and a window displays like this. We could sketch this molecule using the templates here and the bonds, but we have a chemical dictionary which is quite useful. So we're just going to type the name of that inhibitor. Let's show them. Go here. We can look at we wanted to look at the properties of this molecule, if we changed it, these properties would change on the fly. It flags the beta-lactam group and other regions of the molecule if you're interested in looking at that. Okay. And then we've sketched it, so now we can put it into a chemical spreadsheet. So we just click on the chemical spreadsheet button here and just give it a name. Go OK. So now we should see, we can close this, it doesn't matter. Now we have our ligand to dock shown here. So simply to dock it, we go to docking and we choose the option dock. In our case, we're going to dock the chemical table because we built the chemical table shown here. Or you could dock an SDF file that's stored locally on your machine. You go to set up bats from SDF. Or you could read the 3D molecule into ICM Workspace and dock that. But in this case, we're docking it from a table. So go dock chemical table. And we choose, we only have one table, so it finds it. And we'll choose the option dock immediately. Okay, so let's let that run. You see it's running here. It's telling us there's one background job running. It's docking. And so while it's docking, we can look at the other inbuilt reactions that we have in ICM. So we go to File. It's actually in your distribution. So you'd go to Program Files if you're on Windows, MolSoft, ICM-Pro, Covalent Mechanism. It's located there, or in your ICM Home on Linux. Basically, it's in the location where your distribution files are for ICM. So these are all the inbuilt reactions that are already pre-drawn. And so you can sort this table by residue that it interacts with. So number of cysteine reactions, and also serine. And there's also comments on the targets and the name of the kind of reaction that's being done. So you can use that. You can search this table for one that's to see if your reaction is there. If it's not there, I'll show you how to sketch your own. So now the docking is finished. We just go OK, and we see that this was the table we docked, and these are the results. So we have the first hit is ranked by score, which has got a very good ICM score of -47. So we can just click on that and display it. And you can also maybe toggle the other conformations in the stack just by clicking on these buttons here. You can change the visualization. I'm just going to try and change it to what we call pretty. Get rid of the spheres. So, okay. So you can see that the dock ligand has created a covalent bond with the serine here. And it might be interesting to compare it with the crystal structure docked ligand as well. So you can do that by clicking here, and we can see that the redocked and the docked. Let's just change the color of this one. This is the crystal structure one. You can change the color of the carbons only to green. Okay, so you can see it's redocked very well. We could even calculate the RMSD. I made the selection here for the crystal structure. I can make it into another selection called orange selection. So we have orange crosses, and then select the docked ligand in the docking receptor here. And then we can go Tools, Analysis, RMSD, choose the chemical. We're not comparing proteins, we're comparing chemicals. So go to Chemical. We want to compare the static RMSD, and you go Apply. So the first molecule will be the orange selection, and the second one will be the green. And then you see it reports the RMSD of 1.1 angstroms, which is pretty decent. You may improve that if you ran the when you dock it, you could redock it, uncheck here, and then increase the thoroughness maybe to maybe 5 or 10 or something like that. That might improve it, but overall it's pretty decent redocking. I had a question about what other residues can you choose for covalent docking. Basically, you can see in the table we have in the inbuilt table, the covalent mechanism table here, we have serines and cysteine reactions, as well as I think threonine as well. One for threonine. You could choose lysine, but you need to sketch it. Any other ones that aren't included. So any nucleophile that could make a covalent bond you could include. So which is actually what we're going to get onto now. So how would we go about sketching this reaction in ICM if you don't have a reaction in the pre-built example? So we need to just go here, sorry. So to sketch the reaction, we need to open that ligand, the molecule editor again, this one. That's going to undisplay the properties and delete the molecule. Go close. So now we can sketch the reaction. So I'm just going to actually reproduce that one we just used. So it's like it's the reaction whereby you an isolation of the serine side chain by beta-lactam. So we need the 4-membered ring here, and then we can use the bonds to sketch like this, and we have we can use the keyboard for some shortcuts as well. Just quickly, then we need to assign the R groups. So this is the for the as shown, and this is the beta-lactam ring here, and so we need to assign. You've got the oxygen off here, so we need to assign certain R groups that can change on each point. So this one would be R1, R2. You right-click on the atom and choose the R groups here. 3. And then we draw the products as well in a minute. So you can also play around with the stereochemistry. If you right-click on a bond, you can chase stereo down, up. And then for the reaction, you need to separate the reactants and the products using this reaction arrow here. So you just click here, click anywhere. Now we need to sketch the product. So we have the, sorry, just bear with me for a second. This is the oxygen and the serine, which is going to be modified, and then we go down to the rest of the structure. So we have the R5 here, R4, so, and then another bond here. This is the nitrogen with the N1 attached, with the R1, sorry, attached. And this is the carbon with R2 and R3. Now we need to build onto this serine. So this is where the covalent bond occurs. So we have the oxygen of the serine, and then the serine has the C beta and C alpha here. Okay, and we need to tell ICM that this is the product, and this the labels of these atoms need to match the labels of the serine. So let me just undisplay. Okay, so the serine is here. So you have the C beta here, C alpha here for the serine. So you right click on this atom in your sketch and you choose edit atom label and tell ICM that this one is CB, carbon beta, and this is C alpha. So if it's a lysine or something, you'd have a longer chain and you'd need to label accordingly. If you look in the covalent mechanisms for things like the cysteine, you also have the sulfur, and then here, it's a similar thing. So now we've made the reaction. We just need to save it to a chemical spreadsheet as we did before. Just click here. We call it anything you want. Let's go OK. And then the reaction is saved here. So then to use that, we simply go, when you go to docking, you just need to go to the docking menu, choose Covalent, and we have the original one that we used. We can click on the row and delete that one. Okay, yes. And then the source for the reaction library, rather than the inbuilt covalent mechanisms, we can use our own one that we just sketched. It's here. So we just click on it and say add to list, and then it's ready to be used. Just go OK. If at any point you want to turn off the covalent docking, you can come back here and it will turn it off, and then remake the maps for the serine, and you can just do regular docking to your structure. Okay. So that is the basis of it. If you can obviously do screening, so you could have many hundreds of compounds or thousands with so long as it matches the reactions that you have in that you selected in the docking menu. Okay. So you can also use the ICM-3D fully interactive ligand editor. So this is designed for when you have a lead compound and you want to improve the activity or selectivity of the molecule. You can do it interactively in this environment where you can change the ligand in 3D or in 2D and see the effects of the changes on binding. And we'll have a webinar on the whole editor on October 11th. It was developed in close collaboration with medicinal chemists at Novartis, and you can read about this along with other tools that we developed for Novartis in this paper here. So just have an example for the ligand editor. So if we go to open ICM, it's going to just going to get a new session, completely new session, and we're going to look at a cysteine protease called cathepsin S. It performs a fundamental step in antigen presentation, and so therefore it's a good drug target. And the code is 1MS6 in this example, PDB code. And you have some, we can look at the interactions. If we right-click on the ligand and choose ligand pocket interactions, and then just zoom in, just by double-clicking on the ligand, we can zoom in. And it has like a dipeptide nitrile inhibitor. The nitrile's here. And it makes a disulfide bond, covalent bond with the cysteine residue in the pocket. So you can see the covalent bond here. And nitrile is quite close to it. I had some other questions. I think I have to get to them at the end, or I'll get back to you. Thank you. So the covalent bond is here, and so we can use the Ligand Editor to try and improve this inhibitor, or use make changes and try and explore some different chemistry or some ideas that you might have. But first we need to keep the disulfide bond. But to use the Ligand Editor, we use the Ligand Editor tab here. Click on the first button after the ligand, go OK. And then we need to tell ICM which is the receptor, so we just work our way along the buttons. The next button is set up receptor. And because there's only one receptor, it finds it straight away. It's 1MS6. There's different options for waters, which we've discussed before, and optimization. We just leave it as it is. We could use ICM Pocket Finder to find the pocket, but in most cases in a Ligand Editor, you'll have a ligand bound or a complex, and you just say make box around existing ligand. We have the purple box here, and you can drag it as before. Just go OK. So we're making maps, and you can see just by the nature of ICM and most other software, the covalent bond is broken. So we need to restore it. This is the cysteine here, and this is the nitrile. So we need to restore that covalent bond in order to modify the ligand. So we need to actually get a hydrogen. So we need to obtain a terminal hydrogen to define the connection point between the ligand and the receptor. So to get a terminal hydrogen, we can modify this this bond. So we can click here, and we can change this from triple bond to a double bond by clicking on the double bond here, and then single click on the bond. Okay. So you can see we now have a double bond at this position. And this is the cysteine that we need to make the disulfide bond to. So we then select an atom in the ligand, the terminal hydrogen that we just created. Click here, and you can then choose any atom in the receptor, on the cysteine in the receptor. So we can just choose here, and we go to the advanced option and choose define covalent ligand. And it tells us what we just done to do what we just did. Pick a hydrogen in the ligand to attach the modifier. Pick any atom in the modified residue. And go OK. You can see now the disulfide bond is created, and it's building the maps. And you can see the VLS score, a prediction of the score is calculated as a function of a number of different grid energy maps that we calculate. And so the lower this score, the better. Which means that you can do docking. You could redock it. It's going to change the view again. Go to preview. You can redock it or make modifications. Just go back here. So this is the ligand that we'd modify, and so you can then use the Edit Tools panel. You can make modifications like this, undo, redo, and the disulfide bond will be maintained. You can also run the docking or minimization options here as well, and we were covering all of that in 2 weeks' time. So on that note, we had a question about, can ligands to coenzymes be covalently docked? Yes, they can. You just need to select the coenzyme in the same way, basically, with the docking menu. Define which is the atom that's going to be covalently. You define the nucleophile and then sketch the reaction and dock as well. So there are some other questions which I'll get to later on, maybe via email, or just want to thank you very much for attending the webinar. I appreciate your support and for your questions. And if you have any questions after the webinar, please feel free to ask now or via email. Next week, we're looking at fragment docking, screening and linking, followed by the 3D Ligand Editor the week after that, and then moving on to away from crystal protein structures and into the environment of ligand-based fields, 3D pharmacophoric fields, using the method called atomic property fields. Had a question about how to do virtual screening using this approach. It's exactly the same method. So you would just go to, you just have a larger, let's go back to the original beta-lactam example. This is the beta-lactam. You would have a database of potentially covalent drugs, or here you could load them in as an SDF file, go File, Open, and dock them. But you need to make sure that you have the reactions that match your ligands. So you'd have to double check. You can have more than one reaction in your reaction table, and you can then go to docking and then set up batch from a file, SDF file, or from a table loaded in ICM. And then you would get as we got here, you'd have the score, and you can sort the score and plot and select and other things. We had a question about the command line. Yes, everything is everything you do in the graphical user interface you can use in the command line. So generally, it's best to maybe set up the project. You can automate the setup on the command line if you wanted to, but generally it's best to look at the pocket in the graphics and set everything up that way, and double check the charges and your receptor. But if you want, but once everything's set up, I'll just show you. So once everything's set up, you have a number of different files. So you have the grid maps here, the receptor object here, with the name of the docking project. So my doc. You have your reaction here as well that you're using for the covalent docking, and you have the table. This is a table file, so you can change parameters, including the SDF file that you're going to dock. And once you have all these files set up, you can then go to the command line on Unix or in Windows and use the program called, let me just find it, DocScan. So go here. Just going to try and find the help for this. So on the command line, then you can run it like this: the path to ICM, the path to the program DocScan, then the directory with the project name. So in my case, it would be it's a bit tricky in Windows, but it's like C drive documents or what have you. And then you've got a number of different parameters after that you can choose. So if you're doing screening, you could say you can have dash from compound number 1 to 50,000, or from 50,001 to 100,000, whatever. And it will know you're doing covalent docking because you've already set it up that way. It has the reaction there as well. So that's in docs. This is DocScan. If you're using Windows, it's easier on Linux, obviously, but on Windows you can run it on the command line as well. If you go to our website, and go to the knowledge base, if you just type some search string here, that's DocScan. So I'll tell you how to do it in Windows. Didn't like that search. But anyway, you can search for the instructions on Windows or in Linux. Windows command line tips. Yeah, just gives you the best way to run. So here's the DocScan, for example. You'd have the path to the ICM console 64.exe DocScan, name of the project, and then the arguments. Had some other questions. I'm going to have to get back by email, I think, because they're not easily answered. I had another one. Could template? Yeah, yeah, that was another option. Had a question about template docking. Yeah, if you have a group that you know should interact with the serine or something, like in this example, you could just extract the 3D template of that region. So just do that. So I can extract this. I was going to clone this molecule. So I right-click, say clone, and that makes a copy of that molecule. I don't want the receptor, I just want the ligand. So this could be our template. If you right-click on there and say template, then we can modify this template. We can right-click and say edit compound, and then delete the regions that we don't want, and just be left with a template. Let's get close. And then our template molecule will be in the ICM workspace, and then we can use the docking dock from template. So you go to docking, and then you'd use the option template and select the template that you just made, this smaller substructure, and just choose substructure matching or atomic property fields, which we'll discuss later. I mean, that would put it in the same environment. Yes, it sort of mimics the covalent docking in some ways. But if you do have the reaction and everything, it might be best just to use the method we showed today. Had a question about the visualization, because yeah, you're right. The transparency was pretty bad. So if you don't like the transparency, you can go to the lighting tab and choose you can drag this toolbar along. For some reason, my default was set to pretty high transparency, so you can change the transparency there. That's just in the docking menu. You had a question about the post-screening analysis. Yeah, so I don't know if you attended the VLS webinar, but there are a number of options here for post-screening analysis. So you can run the screening of a large database and then go through each ligand in the results and, for example, select those and make a hydrogen bond with the particular residue or atom like that. Okay. Yeah, the other questions I will get back to you by email. Yeah, thank you very much for your time, and hopefully you can join us next week for the fragment docking and screening and linking webinar. Thank you.